So welcome to the GMRI. I'm Sam. I'm a final year PhD student and I'll be taking you on the lab tour. In order to look at tissue in the microscope, we need to be able to shine light through it because we see with light. In order to get light to go through the tissue, it needs to be very, very thin. So thin that you can see through it. And in order to get it thin, we need to cut it very thin. The first step in that is to embed it in paraffin, which is kind of wax. So if you come with me, see in here in this machine does our paraffin impregnation embedding for us. So paraffin is a wax like a candle wax. And you can see in the inside there's a chamber that we put our samples in, and then that cycles through the different chemicals. So that is a chamber where the samples go in there. And then down the bottom here, we've got different chemicals and wax that goes through it. Once we have our sample in wax, we can then embed it over here. So this station here has got a warm part and a cold part. The warm part is to keep the wax liquid, paraffin liquid. The cold part is to freeze it into blocks. And then I'll just find out one for you. So we've got little molds in here. We put the tissue in there, and this is a dispenser for wax. So just like your soft drink dispensers at your fast food, but it dispenses wax rather than soda. I've got the liquid wax sitting on the top there. The blocks, after we've embedded them, come out looking like this. I'll show you these guys here. Find a nice one. And so you can see the tiny little pieces of tissue sitting in there. Because it's hard in this wax, this is literally the same wax that you have candles made out of. Because it's so hard, we can slice it very, very finely, very thinly. And that's what we do on this machine here. So underneath this red guard here is a razor blade. Very, very sharp. And when we spin this handle, it moves the block up and down. What you can't see is it actually moves it along very slightly. And it moves along by 4 micrometers every slice. So that's how thin we cut the tissue. If you think of the difference between a millimeter and a meter, right? The difference between a micrometer and a millimeter is that same difference. So we cut our sections to 4 micrometers. It's thinner than a strand of hair. After that, we put the sections on glass slides like this. You see these slides are all prepared, ready to stain. Because we've cut it thin, you can see through it, but you can't actually see anything because it's all transparent. So after that, we need to actually stain it to give it color. And so we have this robot here, which does the staining. This arm lifts up these racks that we put slides in, and it dunks them into different kinds of dyes and different washes that we use to be able to see our tissues on the slide. We also have a special kind of stain. This robot here is our auto stainer. So this does our special stains. So when we're interested in a particular protein, for example, a particular component inside the cell, we can do very specific stains for those particular components using this machine here. If you want to get a little bit more technical, we can say that those are antibody-based techniques. So our bodies produce proteins that, say, bind to a specific bacteria to target infection, they bind to specific viruses. We can engineer those antibodies to bind to specific targets, specific proteins of interest, and then we can tag a little color onto that, and that then allows us to see those specific proteins in our slides, and that's what we do in this machine here. Next, we're going to have a look at our confocal microscope, which is where we look at these slides. Follow me. So, this is our lovely confocal microscope. I will reveal it for you now. We keep it specially covered up because this is probably the most expensive piece of equipment in the entire lab. One of these tiny lenses in here, have a look in there. You see the lens in there? One of those lenses costs more than any other microscope in the lab, and we have quite a few microscopes in this lab. This microscope sits on a floating air bed with a slab of marble to protect it from vibrations, and it is very, very sensitive. We use it to get very, very high resolution images. So when we're thinking about images and microscopes, you would imagine that actually zooming in would be the most critical part. Getting the biggest magnification is important. But at a certain point, you also want to get resolution, so how clear the image is. And this microscope lets us take very, very clear images. How it does that is a few special tricks. So firstly, it uses lasers rather than an ordinary light bulb. So most of this microscope is actually hidden behind the bench here. You can't see it. We've got some control boxes down here, some computers. That is actually a big component of the microscope that you don't even know is there. So laser light is very focused compared to light that you would get from an ordinary light bulb. That allows our image to be more in focus as well. The other difference, and this is where the term confocal comes from, is that it has a tiny pinhole. So with our ordinary microscopes that we use light and lenses, a lot of what you're seeing is actually out of focus light, because the in focus and out of focus light all gets past your eyes. What the pinhole does is it blocks everything except for the in focus light. That's one of the other ways in which this microscope gives us really, really clear images. The other really special thing that you can do with that is that it lets you take pictures in 3D. So we can change the focus point of where we focus, and that allows us to reconstruct a three-dimensional picture. So we think of microscope slides as flat, but especially with a lot of the work we're doing here with organoids, which are actually three-dimensional, because remember, our bodies are three-dimensional, so if we want to model the body systems, they should be three-dimensional as well. We can actually image them in 3 dimensions as well by scrolling through the focal planes of our 3-dimensional section. The last special thing that this microscope does is it lets us separate out colors. And so with an ordinary microscope, if you're doing multiple stains to look at multiple different proteins, multiple different targets at once, what you get is exactly what you get if you give a child a bunch of felt tip pens and get them to color things in. They're going to use every single color on top of it, every single other color, and it's just going to end up black and you can't see anything. This microscope lets us separate out those colors and look at them separately so that we can look at multiple things on the same slide at the same time. Turning this microscope on is like turning a spaceship on. I've been using this for years now, and I still need to use this to remind myself the exact sequence of how I turn things on. So, go ahead and do that now. So that. left. If you think about a camera in our phone or an actual camera, you have an area which is the sensor. And if you have a 3 megapixel camera, that's 3 million individual pixels that you're capturing on that space. This microscope has that same area dedicated to a single pixel at a time. And what it does is it has mirrors and actually scans across the image, capturing 1 pixel at a time. The advantage of that is that because you're dedicating the entire area to 1 pixel as opposed to splitting it up across your millions of different pixels, is that you get much more sensitivity. So because you're dedicating all of that area to 1 pixel, you get much improved sensitivity. So next up, we'll head through to the main lab. Okay, so what we have here are some storage, essentially, for a lot of our reagents, chemicals and things we use. So these are like exactly the same fridge that you might have at home. The difference being it has a biological hazard sticker on it. You should not store your lunch in here. You also shouldn't eat anything from this fridge. So inside, we keep all sorts of things. So this sort of lemonade-looking stuff is what we feed our cells that we grow. So in our bodies, we have blood that has all the salts and sugars and nutrients that our cells need to thrive and be happy. And when we grow them in the lab, we need to have a supplement for that, a substitute for that. So that is what we use that for. Over here we have our freezers. So these are at, fridges are at 4 degrees Celsius. These are at minus 20 degrees Celsius. And so in here we store more sensitive chemicals, additives, hormones, things that we add to our cell cultures that are a little bit more sensitive to temperature. So that is the minus 80 freezers. In order to keep our cells alive, we need to go even colder. So in the science fiction movies, where they freeze people and then revive them 10 years later, we do that with our cells, because our patients just across the road here get operated on when they need to be operated on. When we want to do an experiment, we want to do 10 samples at the same time so that they're consistent. So we need to have a way to have all 10 samples at the same time. And so what we do is when the samples come in, we freeze them as individual cells, and then when the time is right, we thaw them all at once and bring them out together. So that is over here. There's this freezer here with the biggest hazard stickers on it, because this freezer is at minus 150 degrees. So that's very, very cold. Definitely want to be wearing gloves for this one. This here is a vacuum centrifuge. So we've got a vacuum pump here. This is a machine you can see through the window. It's got little slots and it spins around and around very fast. We use this to dry out our samples. So when I prepare my proteins from my cells, it comes as a liquid, but I want to concentrate it down. So what I do is I dry it out, and then I can make it up again in a smaller volume of a specific buffer, specific liquid that I need for the sample prep. So that's what we do here. Through here, this is a robot that prepares DNA and RNA samples. So lift up the lid, have a look inside. And inside this is just like a miniature lab. So we can put our test tubes in here. This one can shake it to mix them up. This is a little centrifuge, so that spins them down. And then in here, we have got the pipette tips. I'll explain the pipette actually as well, because that's something where we assume everyone knows what it is, because it's very day-to-day, very ordinary for us as scientists, but people who aren't in a lab might not know what it is. So as these things around here, you might have seen them sitting around. And so what this is this is a device that lets us manipulate tiny, tiny volumes of liquid. So if you think about, again, the difference between a liter of water and a milliliter of water, we're dealing in microliters, which is, again, that same step down. So these are fractions of drops, really, really tiny amounts. This is our biggest one. This one goes up to 5 milliliters. That's the biggest volumes we really use in the lab. This is our smallest one, and this one goes down to 2.5 microliters. That amount of liquid is so tiny you can barely see it. So this is liquid nitrogen, so that is, I believe, minus 293 degrees Celsius. So that is even colder than the minus 150 degrees that we have over there. We use the liquid nitrogen to store samples, and we use it to freeze things very quickly. Over here we have our fume cabinet. So some of the chemicals we deal with are a bit dangerous. We don't want to breathe them in, or we want to protect from things falling into it. And so this cabinet allows us to work with those chemicals safely. This is our McIndoe machine. So we sometimes need things to be very pure in the lab, because a trace of anything can throw an experiment off, including some of the minerals and things that are dissolved in ordinary tap water. So this is essentially, it's a very, very high purity filter. So it's similar technology to what they use to turn salt seawater into fresh water. It filters everything else out. And so we use that to make very, very pure water for our experiments. In this room here, this is our autoclave room. So we deal with things that are living cells, living bacteria, and we don't want to release those out into the environment. So anything that leaves the lab, we have to ensure that everything is dead. The cells are dead before they leave the lab. And so what we have here is an autoclave, which is essentially like a pressure cooker you would have at home. Again, you could probably do a nice roast in here, a nice slow cooked casserole would work very well for that. But lab safety, you're not going to do that. It's subjected to very high temperatures and very high pressures, and that kills anything that leaves the lab for rubbish. We have another autoclave here. So this one is a little bit smaller. Inside here, we clean anything that gets used for cell culture. So when tissues come in, we use scissors, we use scalpels, we use forceps and tweezers to deal with the tissue. And the one component that is sort of missing from our tissue that we really can't really replicate in the lab very well is the immune system. And so the conditions that we keep our cells under are also perfect for bacteria. And bacteria and viruses love that media, the liquid I showed you before in the fridge. And they will grow very happily in that. They will outgrow our cells. So anything that we use has to be completely clean, completely sterile. And so we sterilize the water that we use in cell culture, we sterilize the utensils that we use in cell culture. And that's what we use that one for. So this is where we prepare our samples to look at the proteins present in them. I'm particularly interested in proteins for my project because while the DNA and the RNA are sort of the blueprints of the cell, the protein describes what it's actually doing. The proteins are the ones that do all the work inside the cells. They describe what the cell is, how it functions, what it's doing. So this is where we prepare our protein samples. So we have got our test tubes in here. We've got our pipettes. So these are for measuring tiny, tiny little volumes. We've got all of our buffers and solutions and chemicals that we use to prepare the samples. Some of the things we do in here are we run protein gels. So that is what we have in here. So probably the easiest way to describe that is if you take a pen and you touch the tip to a piece of tissue paper, you get the ink bleeding out in rings. And if you have specific colors, you can actually get separation of the different colors in the ink. When we have a mixture of different proteins, we want to separate them out so that we can see the individual proteins rather than the mixture of everything mixed together. So what we do is we load our proteins at the top of the gel. You can see the little indentations in there. We load our samples in the top, and then we use these power packs here to run an electrical current across the gel. The electrical current separates out our proteins, and they travel differently based on their size. So that lets us separate out proteins by size, which is one of the ways we analyze proteins here. This machine here is got a camera up the top here, and inside here is a bed where we put those gels on, and then we have special illumination, and that allows us to then see those different bands of the different proteins. And that's the main thing here. This particular lab has got an extra warning sticker on it. If you were here in person, I would remind you to not touch anything in here, because this is possibly the most dangerous lab that we have. But again, you're safe at home, you won't be able to touch anything, so come on in and have a look. Okay. So in this lab we produce lentiviruses. And that is why this lab is separate from the big cell culture lab. So we have the same equipment in here, it's just smaller and cleaner. So we have got here the centrifuges, which I use to spin the samples down and separate them out. I have a microscope under here to look at my cells. I have got a water bath here, which we keep at 37 degrees Celsius. So our bodies are at 37. Our cells like to be at 37 degrees Celsius. All our salts and solutions and things that we use, we keep at 37 as well to keep our cells nice and happy. This here is the incubator. So it's not running at the moment. That's why you can't see anything on the screen. How I describe this is, so we're interested in human cells, and our bodies actually do a lot that we just take for granted. Some of the basics are keeping us at the right temperature, keeping us at the right humidity, keeping our acidity levels correct. And so all of that we need to mimic as closely as possible. And so inside here, this is heated to 37 degrees. And it's humidified, and we make sure that we keep the pH controlled, we keep the carbon dioxide level controlled, to mimic what we have in the human body. One thing that we can't replicate in the human body is the immune system. And so the consequence of that is that we need to keep everything sterile that we use to culture our cells. So the liquid we feed them has to stay sterile, the equipment has to stay sterile. Our hands and arms, we spray those down with alcohol before we touch them to make sure that that stays sterile. And we do the work inside these biosafety cabinets. So there's glass down at the moment, but that comes up, And then it's got an air curtain. So similar to the shops, when you walk into those shops and you get the blast of hot air, that is to prevent the air inside the shop from going out and the outside air from going in. And so this does two things. So it protects the cells from us, from me, because I'm carrying bacteria everywhere and I don't want to get those into my culture accidentally. But also, if I'm working with viruses, for example, that cause cancer and make cells glow green, I don't want those coming out into the environment. And so this biosafety cabinet protects anything from going out and anything from going in. So thank you for coming along on our tour, and I hope you found it interesting.